It is such a tremendous pleasure to be back here. I love this place. This is such a fantastic program. So you're all blessed to be here. And it was an honor as well. I was really surprised by the invitation, so thank you to those of you that invited me. That's the title of my talk. Oh, actually, I already went past the title of my talk. What I'm going to talk to you about today is first introduce the idea of heterogeneity. What is heterogeneity? Why does it matter? I'm going to introduce this idea of neighborhood models. I'm going to want this. Looking at the individual tree scale and trying to understand the importance of a tree's neighborhood, looking at different studies and different places. And then I'm going to try to start to scale that up to the stand. How can we use neighborhoods to understand the impacts of heterogeneity at larger landscape or stand scales? And then I'm going to end with sort of a digression. I got the sense that there were some people interested in hearing about this. So I'm going to talk a little bit about the path that I took, the type of school that I'm at, and some of the choices that I made in case it helps you in any way. Is my volume OK in the back, by the way? OK, great. Thank you. So does this also advance? No. OK, good. Just checking. Good. I'll just wander. So forests. Forests are really complex systems. They vary spatially and temporally. Heterogeneity, the word I'm using, has become a real recognized buzzword, I think, for ecologists and managers alike. What is heterogeneity and complexity in natural systems is sort of where I want to start. There has been a bit of a paradigm shift recently, and recently in forestry would be the last 10 to 20 years. The idea of ecological forestry, okay? This recognition that heterogeneity is a really important fundamental part of forest systems. It was interesting, for those of you that were at ESA, some of you might have heard Jerry Franklin talk, and there was also an entire set of talks that were dedicated to his work and his legacy. And what I really took away from that was that one of the big things he left us with was this idea that management of forests really needs to embrace and emulate natural disturbance processes because they increase heterogeneity in systems, and that heterogeneity is something which really maintains the ecological integrity of forests. So what does heterogeneity look like? It's hard to wonder. This is not designed for wondering. I'll illustrate with a super oversimplified cartoon, stand development. Basically, we start with a somewhat simple structure, and through natural disturbance processes, mortality, self-thinning, fire, wind, disease, we change the character of the stand over time and ultimately arrive at something which is more complex or more heterogeneous. Those changes can be episodic, they can be gradual, but complexity, okay, is this sort of thing that we hope to achieve someday. What does it look like? You can have complexity in structure, the spatial and vertical arrangement of trees. You can get more size differentiation. You can get stratification of the canopy, the opening of light gaps, changes in microclimate and regeneration niches for seedlings, etc. We know that all of the changes happening in the vegetation are paralleled by equally important changes, which I'm not going to discuss, in microbes and fungi and birds and animals. All right? So these are all really important processes. How do we describe that? How do we describe heterogeneity? I would argue that from the perspective of an individual tree, which is the fundamental unit of interest, if you're a forest ecologist wanting to understand growth or regeneration or survival patterns, we're talking about an individual tree. And for an individual tree, heterogeneity means that not all trees are experiencing the same thing. So in a really simple stand, like this one, all of these trees are sort of having the same experience. This is our forest cult, if you will. And in an old growth forest, you have something different. All of these trees are not experiencing the same thing. They might be experiencing very different resource supplies, above ground and below ground, different amounts of growing space, different biotic interactions. And so what I'm going to try to convince you of first in my talk is that the neighborhood matters, okay? And the neighborhood is this space over which an individual tree is interacting abiotically and biotically with its resources and its neighbors, okay? Trees are locavores. They don't move, and they operate over a relatively small spatial scale. And it is that scale that I think is fundamentally important for tree processes like growth. An individual tree really depends on both the structure and composition of its neighborhood, which I'm going to walk you through. But then I would argue, this is my new perspective, developed over the last 2 or 3 years, that we can scale up those processes beyond the neighborhood to look at stand-level patterns. And the argument would be that if a tree depends on its neighborhood, I believe a forest is a collection of individual trees. I don't really see evidence of emergent properties in forests. If an individual tree depends on its neighborhood and we have a collection of trees, then I kind of look at a forest as being a big collection of unique ecological neighborhoods, and that's what we should be thinking about. And so heterogeneity is really a change in the distribution or frequency of types of ecological neighborhoods on a landscape. And I'm hopefully going to spend the second part of my talk showing you how I'm attacking that question. So if we want to describe a tree's neighborhood, starting at the lowest scale, how do we do that? I'm going to really gloss over this one. We map the trees, we know where everything is, and am I projecting? I'm clicking. OK, well, here's our focal tree. And what we're saying is that a tree's neighborhood, all right, or the amount of competition it has, is going to depend on the biomass of the neighbor, the size of that neighbor, B, divided by the distance to that neighbor. So if a tree is further away, it's going to have a diminishing impact. And we're going to calculate that for every tree within a given radius and sum it all up, and we're going to get this nice little number here that we can plug into any sort of modeling or equation that we would like to invent. And that's going to be the neighborhood index.
So there's a laser pointer on there.
Is it just the button on the bottom?
There's a little button on the top that says laser.
Where? There we go. Perfect. Thank you. And so we get back this great number. Here's an example. This is the neighborhood index down here that we've calculated. And I'm looking at the relationship with growth, tree growth. And I'm seeing that, in general, growth is declining as you increase the index. Now, remember what that means. That can be an increase in the number of trees around that tree, the size of the trees, or a decrease in how far away they are, the closer the trees are. And if growth declines, that suggests that competition is occurring between these trees. All right? So the next question is really, how can we use this? Why is this of value? What can it tell us about the important attributes of a tree's neighborhood that it's really responding to? So I'm going to walk you through a couple older case studies. Actually, this one's brand new. The first thing that I think is really important in a neighborhood is density. This is actually not using the competition index, but it illustrates my point pretty well. This comes from some work in Minnesota, and it comes from some pairwise trials of seedlings that we monitored over 10 years. And so we're looking at competition between larch and black spruce, larch and white pine, black spruce and white pine. Don't really worry about it. I'm just going to show you one thing. We're looking at diameter and height, and we've planted them at a range of densities, 1, 4, 16, and 64 trees per meter squared. So we're really trying to get at competition as a result of density. And what's interesting is that changes in density alone actually can lead to rank reversals, which is really an interesting topic when you're thinking about stand development. Okay? We're actually changing the outcome of who wins during that early important stage of stand development. So when planting density is low, Pinus strobus is on top significantly. Okay? It's got bigger diameters, and it's got equal heights. But if you increase the density, you're actually going to see a reversal, which is significant. That's what that statistic is telling us. And out here in a dense neighborhood, the larch has significantly larger diameters, and down here it has significantly taller heights. So really my point is just that competition really does structure the outcome and can really influence potentially the trajectory of a stand at this early stage. So density matters. What else matters? Species matter. Trees do care about the identity of their neighbors. This comes from Hawaii, work with Dan Binkley and Randy Seenock. Lots of other people have worked there. This is a replacement series. We have Falcateria, which is a nitrogen-fixing tree, eucalyptus, which is a fast-growing tree which tends to be nitrogen-limited. And what this means is we have plots with 100% Falcateria, 100% eucalyptus, and lots of combinations in between. Makes it a good place to study competition. These plots also occur on a nice natural gradient of soil nitrogen and soil phosphorus, which is going to lead to another question in just a minute. So what do we find? I sort of cranked out this sort of new way to look at these data. Species identity really matters. So this is looking at the effect of Falcateria neighbors through the neighbor index on changes in growth, how it's affecting growth for eucalyptus as our focal tree in red versus Falcateria. So what we see is that Falcateria trees are competing strongly with one another. Intraspecific competition is very strong. It reduces growth by 80%. Okay? As you increase the number of Falcateria neighbors, you're reducing growth up to 80%. On the other hand, Falcateria trees are not competing at all with eucalyptus. There is no competition going on there. Actually, it looks like there's a tiny bit of facilitation, which we'll get to. So species matter. But what's even more interesting is that the interactions between species at this neighborhood scale really do depend on soil nutrients. And I found that over and over again in my work. There is some rich history there. If anyone pays attention, there's constant debate about whether or not competitive intensity between plants should increase with soil fertility or decrease with soil fertility. And it really goes to the heart of sort of what structures communities and how important competition is. Grime, with his CSR strategy, originally proposed that competition should increase with soil fertility. And Burtness and Callaway sort of supported that with their stress gradient hypothesis more recently. Tillman really proposes the opposite. He says that competition for soil nutrients should decrease as nutrients become more abundant in a system. And that's the resource ratio hypothesis. So what did we find? We find that species matter, but that depends on nutrients. So this is looking at the effect of Falcateria trees on eucalyptus growth, just eucalyptus, so interspecific competition for trees on high phosphorus soils in red or low phosphorus soils in blue. So we sort of separated our data. And this is a modeled relationship, which is why it's a perfect line. And what we see, I'll stay over here for this one, is looking just at that blue line first. When there's not a lot of phosphorus, Falcateria trees are really competitive. They're reducing growth by about 20%. These are competing. But at high phosphorus, on that red line, we see that those neighbors are not competing. They're actually somehow facilitating eucalyptus, right? As you increase the number and density and size of Falcateria neighbors, eucalyptus trees are actually growing better. Now, the most logical explanation, luckily we have a good explanation in this system, which is that we have nitrogen fixers. And Falcateria fixes nitrogen. They're limited by phosphorus. Most nitrogen fixers are. So in a high phosphorus system, they're getting more phosphorus, they're fixing more nitrogen, they're adding it to the soil, and the nitrogen is helping out their euc neighbors who need more nitrogen. But on a low phosphorus soil, they're not fixing nitrogen, or very much nitrogen, so they're not really helping out their neighbors, and they just become nuisance neighbors, which is what most trees are, and they're competing for other things, like light or water. So suddenly we just see a net competitive impact. How about intraspecific competition? Now we're looking at the effects of Falcateria on Falcateria. And we see a very, very different pattern. And what we see, if you look at the blue line first, this is looking at their relationships in systems with a lot of phosphorus. And that now, by now, you see the down slope. That should indicate competition. So Falcateria trees are reducing the growth of their neighbors by 80% when there's a lot of phosphorus in the system. That supports Grimes' theory. Competition is getting much more intense between these species that are the same when nutrients are more available. But with the red line, we don't see any evidence of competition at all on a low phosphorus soil. What's more interesting is this crossover, which I can just speculate about. But if you look here, when there's not a lot of competition in these neighborhoods, when there's not a lot of Falcateria, trees are growing better when they have more phosphorus. The blue line is above the red line. That makes sense. They want more phosphorus, you give them more, they grow more. That's how we think the world should work. But as you increase the amount of competition from neighbors that do the same thing and need the same thing as you, they actually reverse. So that by the time you get out here to a really high neighborhood index, and remember that probably means the focal tree is smaller, it's surrounded by really big neighbors. that tree is performing better on a low phosphorus than a high phosphorus soil. So maybe the benefit on this soil is that they might be getting a little more phosphorus. But the cost of being on this soil is that all of those big neighbors are also getting more phosphorus and are potentially more vigorous and more competitive. And ultimately, that is a bigger cost than a benefit. And so we're seeing ultimately competition on a more fertile soil, which is what Grime proposes. Species matter, density matters, genes even matter. A tree actually cares about the genotype of its neighbor. This comes, again, from work with Dan and Jose Stape in Brazil. You can see these are big plots here. And what we're looking at least in this piece, is competition between eucalyptus trees that either are genetically identical to their neighbors because they have been clonally propagated, or they are genetically variable. Same species, but they come from seed. So we got natural genetic variation. And we're looking at how they relate to one another. And one simple result, we have great evidence that genetically identical trees are competing much more intensely. So as you increase the neighborhood index, genetically identical trees are suffering a 30% reduction in growth overall, relative to trees that have genetically diverse neighbors. That's a pretty, it's easy to accept that. That's something we sort of have seen and recognized, and we sort of like that idea. The fact of the matter is that that sort of pattern hasn't been demonstrated in long-lived perennials outside of sort of experimental plant systems like Arabidopsis, and has never been demonstrated for trees before this particular work. It does support really rich theory. It supports ideas of resource partitioning. It supports the idea that ecosystems should actually tend towards greater diversity. It supports the evolution of sex and seed dispersal. All of those great ideas in ecology depend on this notion that organisms that are closely related to one another are ultimately more harmful to each other and should therefore spread out, right? And that does support those theories. So I want to synthesize this neighborhood scale, which encompassed a little bit of new work and mostly my old work. This is sort of where I started. We saw that density does matter at this neighborhood scale. The strength of competition can lead to different successional paths, I would argue, when you sort of look at it in that stage of development. Neighbors of different species or different genotypes really are not equivalent in their competitive effects on trees. So trees care about the identity of their neighbor. Resource supplies can really affect both the nature and strength of those interactions. Okay? Resource supplies are important. Understanding this relationship with soil fertility is important. Ultimately, I found more support for Grime's hypothesis that trees are ultimately more competitive when they have more of their limiting resource, okay, on a more fertile soil. So let's move forward. Ultimately, the biggest thing I hope I've convinced you of is this first part, that trees do care about the structure and composition of their neighborhood. And now I want to take you to this part two, okay? Does understanding the distribution of neighborhoods at a larger spatial scale help us to explain stand-level processes? And ultimately, can this neighborhood approach help us to understand the consequences of stand-level heterogeneity, which these days we really do want to understand? Some of these emerging practices in ecological forestry that Franklin was talking about are gaining in popularity. You see them more in certain parts of the country. So this is a variable retention harvest. The goal is to create heterogeneity. You change the spatial pattern and arrangement of trees that you're leaving behind. And it looks more heterogeneous. We don't actually have a lot of great empirical long-term data sets that look at the consequences, okay, or ultimate effects of these sorts of treatments on things like resource supplies and regeneration and stand development. We don't really know the outcome yet. So one of the goals of the people the group I was working with in Minnesota, my collaborators were Rebecca Montgomery, Peter Reich, and Brian Pollock. Brian is with the Northern Research Station in Minnesota, was to really try to, at a large scale, ask the question of what the effects of heterogeneity are in forested ecosystems. This is replicated 4 times across the landscape. It's massive. This is being done in red pine forests in north central Minnesota. And the goal here was to change the spatial patterning of trees, change heterogeneity only, and to leave the amount of wood the same. So we want to leave the same basal area, so we have some uniformity, and just change the pattern. In 4 treatment types, we have our control, we have a uniform thinning, think simple, right? We have small gaps, and we have large gaps. Someone who's perceptive might suddenly be looking at that and wondering whether or not we really did leave the same amount of basal area in the forest. I'll address that one later. That was our goal. We wanted to know how pattern, okay, how does the pattern and heterogeneity of trees affect resource supplies and regeneration? At least that's the piece I'm going to be focusing on today. And our hypothesis going into this was that despite constant tree abundance in these treatments, the average resource availability across the stands and heterogeneity within the stands is going to be greater as you increase the patchiness of the cut, or as you increase heterogeneity, okay? And our reason for that is really embedded in this model. And this model is in the paper that is published this summer in Ecological Applications, but it has roots in other similar models from Deborah Goldberg and previous work of Brian Pollak's. And what the model suggests is that resource availability, and I'm speaking generally at this point, is going to decline rapidly, in fact exponentially, as you increase competitor abundance in a forested ecosystem. So you're going to have low resources at a pretty large range of competitor abundances. And resources are really only going to increase rapidly when you get down to pretty low levels of competition. I'm, of course, going to substitute this neighborhood approach in for this competition abundance thing, because that's how I measure those sorts of things. So what this suggests is that if you go in and you thin a forest or manage a forest in some way, what you're doing is you're going to change the pattern of competitors along that x-axis, and ultimately, you're going to really change the distribution and availability of resources. Specifically, a very simple structure is going to create a narrow range of competitor abundances of neighborhood types. And a more patchy or heterogeneous structure is ultimately going to create some high abundance neighborhoods, low abundance neighborhoods. You're creating greater variation. And because this is a nonlinear relationship, you have a greater chance of having some really low competition environments where suddenly you're going to get a disproportionate increase in resource supplies. And that's going to allow your average resource supply to go up. And I'll walk you through that again if that was a little bit confusing. So our objective was to develop these resource response curves for nitrogen, phosphorus, and light as actually measured in the stand, and then see how that relationship I just described, how that relationship is driving changes in the mean and the variation of resources for our stands and for some simulated stands, which was a piece that I added. I wanted to see if we could use this as a modeling tool. So basically, we went out and we sampled everywhere with lots of people for many years, 192 sample points. I mapped all the neighborhoods. We measured light, dawn and dusk for years. We measured nitrogen and phosphorus with resin bags. And we measured all kinds of other things. And I'm going to jump right in. Let's look at what happened to stand-level resource patterns, the first part of our hypothesis. I sort of color-coded this to make it easy, and this is based on a basic sort of analysis of variance model. We've got our 4 treatments: control, dispersed, small and large, light, phosphorus, and nitrogen. And significance aside, you can see in the middle anyway, that the control always has the lowest resource availability in the soil and for light, and that as you increase the sort of patchiness of these cuts, resource availabilities are going up significantly in the large gap. The dispersed and the small are not significantly different from one another, if you look at my letter coding there. They really don't come out, and I'll talk about why in just a minute. But at least at this scale of analysis, we can say, yeah, okay, well, you cut down trees, you reduce plant demand and uptake of resources above and below ground, and so suddenly resources become more available. That is not anything really new. Let's look at the neighborhood scale. Now what I'm looking at is all of our neighborhood indices calculated for all of these points, okay, plotted here. And I'm just looking at light. I don't really have time to do nitrogen or phosphorus, but the same pattern existed for nutrients. It just was not quite as strong. And I've plotted all of the treatments together. And what you see is that when they're all combined, they actually encompass the full range of light conditions. We have a lot of variation across all 4 treatments. And they are fit perfectly, or the best model is an exponential model. R squared is 0.75. What's more interesting is how the treatments break down. So for instance, our control treatment down here has tremendous variation along the x-axis. We actually had a lot of natural structural diversity in these control forests. But there is not a lot of change, pointing this way, on the y-axis. You don't get a lot of variation in light being created because it falls on this asymptotic part of the curve. The curve is not changing down there where our control stands are. Up here, for instance, in the dispersed, you can kind of see these green triangles. We have a narrow range along the x-axis, but we actually get a pretty decent spread on the y-axis. We're getting a fair amount of variation in light despite having a uniform thinning because it falls on this asymptotic part of the curve, which is exactly why understanding the exponential model is so important, is what I tell my students all the time. You can see this a little more clearly when we look at histograms, and this is really sort of where I'm moving towards, the use of histograms. So what I'm doing now is I'm sort of saying, let's look at a stand as this distribution of neighborhood types that I was talking about. And so we're looking at, the values for the neighborhood index and then the frequency of each of those neighborhoods across a given forest type. And we've got, there's a huge spatial area for dispersed, small, large, and control. And then I'm also showing the distribution of light that was measured in those stands. So what was created from that structure. So for instance, let's look at the control again. You have a really big spread, a lot of variation in structure, and really little variation in light. And it's also pretty low light. Up here in the dispersed, you have very little variation in structure, but you're actually getting a pretty good distribution of light environments being created there, which are ultimately going to foster different organisms, potentially, right? If you use the standard deviations of these distributions to compare them, 0.04, 0.21, the control has 5 times as much structural heterogeneity. But it has half of the heterogeneity of light resources. It is not a very diverse environment when you're looking at light. The large gap, you have some open neighborhoods, so you get a little bit more highlight neighborhoods. That's why it stood out in those ANOVAs. Once again, our small gap and our dispersed just didn't fall out. They didn't do things differently, and we really thought gaps should matter. What we discovered pretty quickly is, you can see the mean is 0.14. It really did make a difference. We didn't thin the small gap treatments enough. It operationally did not go as planned, and they just left too much wood out there. So really that pooched the whole hypothesis. It's hard to do large-scale silvicultural operations. You can tell people how you want to cut the forest, but I'm not actually cutting the forest. So what I realized is it didn't really matter. I didn't really care what we had or hadn't done on the ground, because what I was moving towards was sort of a theoretical framework for modeling resource distributions in any kind of forest we might want to imagine. Let's design a restoration program. Let's design a management plan, and let's model what kinds of resources we would get based on the distribution we're going to create. So how do I do that? Well, it's this basic approach again. If we look at a forest, not based on averages, but based on this distribution of neighborhood types, okay? We see a forest as this collection of unique ecological neighborhoods, okay? And that's how we're describing our forest. Then we can model exactly what we're going to find in each of those neighborhoods based on this empirical curve, which is fit to data for our forest type. Obviously, it takes some work. And we can model it pretty well. Our R squared was 0.75. So we can sort of predict what's going to happen in each of these neighborhoods that we've created. So this really gives kind of a theoretical and somewhat practical approach to scaling from the neighborhood to stand-level processes that we might want to predict. And I would argue we could do this for anything. We can do this for production. So what does that look like? We're looking at the exact same layout as before, so it should be familiar. We've got frequencies of neighborhoods here, frequencies of light here, but now these are simulated forest stands. These are not real. This is what I wish we'd done when we cut the forest. And I really took a long time developing these. If we really created gaps the way they should be created, this is what the distributions would look like. You can see that I very tightly control the distributions. They have identical means, 0.25. Average conditions are identical. So if you just go out and look at the averages, hey, these stands all doing the same thing. We've created much greater heterogeneity, 0.06, 0.14, 0.21. And then we model light using that empirical relationship, that curve. And what we see is that even though the averages are the same, we've managed to increase light from 25% mean light, 30 to 35. And those differences are significant. That's what that p-value is associated with. And the heterogeneity also goes way up, 6, 15, 22. So now I've managed to prove or support my hypothesis. You know, it's great if you can't get it in the field, go model it. But quite seriously, okay, this supports the idea. If you have average conditions that are the same, you can actually increase the mean light based on just the heterogeneity of what's out there. And this actually represents a 40% increase in light. So just by changing the patchiness, we've actually increased mean resources by 40%. And what that suggests is I don't really think we can be looking to stand averages when we're thinking about complex, non-linear processes in ecology. And I know I'm not the first one to say that. So the next step was to actually see whether or not things are responding to this. One of the things we were doing is we were planting different species in the understory. And we were really interested in how different species of tree would respond to the heterogeneity we created. Quite intentionally, we planted something which was light demanding, jack pine, red pine, which is sort of an important tree up there in these systems, intermediate, and white pine, which is a little bit more shade tolerant. So we're sort of expecting, oh, this one's going to do great in the big gaps, and this one's going to do better in the uniform thinning, and what is heterogeneity going to do for these different species? You can tell I have a really hard time being behind the podium. So how am I going to get at that answer? This is really new work. Same collaborators as before, but now I really want to acknowledge the involvement of Charlie Canham, who's sort of been my role model for a lot of this work. And he's been working with me over the last year on some of these models. And I also, even though he's not here, should acknowledge Tom Hobbs. Because the first time I did anything like this and realized the value of this approach was in his, what was it called? Ecological modeling. Systems ecology. So bravo. I wouldn't have gone down this path had I not taken that course. So I'm modeling individual seedling growth over time based on all of the data we have. We have light, we've got this neighborhood index, right, the canopy essentially, and we've got nutrients. And these are my models. I'm using likelihood methods and comparing them with Akaiki's information criterion. I can tell you more if you want to ask. So light, these are all nonlinear, and we're adding nonlinear models together. So light growth responds sort of asymptotically. We've got this saturation curve. This is pretty standard for a light response in plants. The neighborhood index is the one you've seen before. So growth is declining nonlinearly with increases in this neighborhood index. There's a C in there. Don't mind the C. The next one is light and neighborhood. And I just want to point out that, the elegance of this approach, in my mind, is that these models are not just, phenomenological, but they're somewhat mechanistic, because you can ask questions by comparing these models to one another. So, for instance, we usually assume that most competitive interactions are indirect interactions for light, right? So you thin a forest, you free up the resource, and it's the change in resource that causes the response in the plant, right? So the way they're the mechanism of competition is really just resource supply. But what we're saying is, well, yeah, let's put light in, which is actually a measurement over the seedling. We measured light right at the seedling, what it's getting. But let's put in the neighborhood too. So in addition to how much light it's getting, does just the structure and arrangement of those neighbors mean something? In other words, is there other processes going on, like water competition, which we didn't measure, or above ground direct interactions, or disease, Jansen-Connell hypothesis? Do neighbors matter in other ways than just reducing resource levels? The next models all added this question that I love, which is, should soil fertility modify the strength of interactions between competing neighbors? So we're going to test those hypotheses again. This model just has nitrogen. This is just testing the role of phosphorus. And this one is actually including additive effects. So it's not a single nutrient limitation model. It's saying, hey, two nutrients can matter to a plant, which actually was not really what we believed for a while. And obviously what these are doing mechanistically in the model is the resources are modifying that rate of decline, okay? How much growth is dropping as you increase competition. And that exponent is being controlled by nutrients. So what did we find? These are our models. You can see here, red pine, the best model, actually included an interaction with nitrogen. These are delta AICs, so the best models have 0, and we're comparing them based on AIC to these other models. 10 or more is highly significant. Differences of sort of 3 to 7 is considered strong support, and you can parse it out. But we went with this sort of simpler model. There's some evidence that phosphorus matters too, but this is a little bit more parsimonious. So this is our best model. Not a great R-squared. We had a lot of disease in our red pine. White pine, R-squared of 0.56. These are approximate. And it's our basic model. These aren't far off. We could argue that nutrients matter, but these days I kind of don't like shoving things in just because they kind of show up on the radar. Are they really significant? Is it really playing a significant role in growth? So I left them out. Jack pine. The best model by far is the most complicated model. This one has a heck of a lot of parameters. And the R-squared is 0.71. And it's saying light matters, neighborhood structure matters, and hey, it's all been getting controlled by soil fertility. So let's look at the data. This is light affecting growth. And what you see is that red pine and white pine really saturate quite early, as you might expect. By the time you get to about 10% light, we're really seeing a saturation in terms of their growth response. Jack pine just continues to respond to light. Even out here where it's sort of in the middle of a gap, we're still continuing to see growth response from this light-demanding tree. All right, so this tells us how they're responding to light in this forest. Just basic story. The differences, I should say, were magnified in the gaps. We saw bigger differences between our species in the patchy cuts and less differences in the control forests. And I just want to point out, because it's kind of interesting, that the biggest response we're seeing, at least in the red and white, is at really low light levels. Okay, that's where you're sort of getting the most bang for your buck, the biggest return, essentially. And really, that's happening, looking back at this model, at pretty high levels of the neighborhood index. What that says is if you're in this type of control forest, it doesn't take a lot of thinning to increase resource supplies enough to really get the sort of maximum response that you're going to get, or at least to get a pretty decent amount of the response you're going to get from these types of species. And that most of our gaps just thinned so heavily that we sort of maxed out what our trees could actually do. This is our competition, okay, neighborhood index and growth. And you see that white and red look pretty similar. Okay, there is evidence that there's competition going on. It's significant. White pine does grow more at every level of competition than red pine. Jack pine actually has a pretty different exponent here. So it's going down faster. It's more sensitive to competition. So in an open neighborhood, it's actually growing quite a bit better than the white pine, all right? Quite a bit better. But by the time you get out into these sort of dense parts of the control forest, jack pine has lost its sort of competitive advantage. Suddenly it's growing about the same as its white pine neighbors. So the interactions. This is red pine. And what we see for red pine is that nitrogen matters, and what we see is the same story as Hawaii. Red pine on a low nitrogen soil are competing. Okay, we got a negative slope, but on a high nitrogen soil, they're suffering more. They are definitely suffering more. You're getting a 30% reduction in growth out here in the denser parts of the forest when the trees have more soil nitrogen. I'm sensing a pattern. And jack pine. Unfortunately, I switched my colors, but let's look at nitrogen, purple. Low nitrogen soils, not a lot of competition. High nitrogen soils, well, their growth is getting reduced from about 17 to almost no growth when they're on a high nitrogen soil. Same story. Same story. But now phosphorus is in green, and phosphorus, the pattern totally flips, which is exactly what happened in Hawaii. So phosphorus, on a low phosphorus soil, we're getting competition, a lot of competition. But on a high phosphorus soil, trees are growing more when you increase the density and canopy coverage from residual trees. Remember where this Ni is coming from. It's coming from the trees we left standing. In Hawaii, we could explain this through the presence of nitrogen-fixing trees, right? Here, we don't have nitrogen-fixing trees, and we've removed the understory to control for seedling growth. It makes me have to wonder what's going on below ground. I can begin to speculate. If anyone has ideas later, tell me. Free-living nitrogen fixers. And what about mycorrhizae? We know that big trees certainly maintain mycorrhizal connections with seedlings. They're planted seedlings, and there's growing evidence that mycorrhizae are pretty sensitive to phosphorus concentrations. So I'm speculating. But it's the same pattern, and I think that's pretty interesting. Same pattern for the same resources. So to summarize, neighborhood structure does affect tree growth and resource supplies. I hope you bought into that piece. Neighborhood structure, okay, neighborhood structure can predict stand-level patterns of resources. But the average structure, those means, could not predict stand-level patterns. Averages didn't really help us out. Developing alternative management strategies requires effectively modeling heterogeneity. There's a lot of interest in being able to do this. I have thrown out one theoretical way to think about how to do that. There's lots of other ways out there. I do like the general model. I think that understanding the shape of the resource response curve and the growth response curves for your system can really help you in designing retention treatments that'll help you to achieve specific objectives and specifically target different organisms that you might be interested in the understory. Consistently, we saw that resource supplies are modifying biotic interactions, modifying competition in communities. And I did see more support for grime consistently, but I would have to argue that I think this debate is kind of meaningless. I think that the role of competition in community processes is really context dependent. So it's going to depend on the species that are interacting. I think you can find other patterns in other places. And it's going to depend on the types of resources that are being competed for. So that's my 2 cents on the debate. I'm going to shift gears, if you'll give me the 10 minutes. We did start a little bit late. And talk about my path at this point. You can save your research questions for just a minute. I think this is sort of one of the things I was asked to talk about. I guess there might be some interest in sort of what I've done. So rapidly, I'll bring you up to date. In the 4th year of my PhD, I had my daughter. It ended up being a really great time to have kids because I only got busier, honestly. And then I went off to a postdoc. Now, I knew going into my PhD that I wanted to teach. I used to teach high school, and I really loved teaching. My parents are teachers. I sort of had no choice. What changed, though, was my growing interest in research, and I really continued that. I love research, but I also love teaching. In my postdoc, I did 2 things that I think are notable, which I'd like to share with you. One was that I asked for opportunities. I had to push for opportunities to teach above and beyond what I was being asked to do for my postdoc, and I didn't get paid anymore. So I taught an upper-level elective, landscape ecology. I designed it, and I taught it on my own, and I did it for 2 years. And wow, did that help me when I went into my first teaching job. The second thing I did is I participated in a program called Preparing Future Faculty. And it was a year-long program that taught me how to teach. I learned about pedagogy. I learned about designing learning outcomes and a good syllabus. I learned about student-centered learning and how to use case studies and a whole lot of things that a lot of scientists don't spend a lot of time thinking about. And I learned it from educators. And I had to watch people in other disciplines and departments teach. I was required to go to lectures every week. And it was mind-boggling what they were doing with classes of 200 people. Breakout sessions in the hallway, problem sets, and I'm like, wow, I didn't learn this way. And it has totally changed my life. And there's a reason why you should do that, which I'll mention again in a minute. And then I have my very cute little son there. And he's 5 now. That's Wyatt. And I had him during my postdoc. So you see a pattern here. I got it all out of the way before I started my job. And I applied only to teaching schools. I really resisted. Everyone kept forwarding me those other jobs, and I just applied to teaching schools. And I went to a state school, 4,700 people or so, part of the state system in Pennsylvania. Not a really strong school, not really high admissions criteria, lots of forests. They wanted a forest ecologist. I get to teach great courses. It really seemed like the perfect job. So before I talk about the job, I want to think for a minute about why this story already made people interested in me talking to you today. So unfortunately, the fact of the matter is that I represent a minority. I represent a minority because I'm a woman in science, and I represent a minority because I'm a woman in science who actually had children. I'm the only woman in my department that has kids. I'm the only woman in my department for the last 30 years that had kids. There is no role model. This is the leaky pipeline. If you haven't seen it, you should have seen it. Probably you've seen it. The fact is that 30% women with babies, 30% likely to enter tenure-track jobs. Women in general, 23% less likely to become an associate professor, 25% less likely to become a full professor. I'm not going to get into all the reasons this might be going on or bash the system. The fact is that this is the system. For some reason, women are not really entering the ranks as they should based on their percentages. This might suggest that some people see family and academia as being somewhat incompatible. And I'd like to broaden that to men as well, because if you want to be an involved parent, no matter what your gender, you're going to have some problems. So most of you maybe are asking yourself, can I have it all? Can I have an awesome job that's rewarding and that I do really well in, but also, be the one that picks my kid up after school, and I can keep cooking homemade meals, and I can be at soccer practices, and can I have it all? And a personal life too, and oh yeah, I'm also gonna keep going to the gym and eating well. This is pretty topical. This was in this summer. I don't know if you saw this article, cover of the Atlantic Monthly, Why Women Still Can't Have It All. Okay, that's her conclusion. It's kind of depressing, lots of outrage on the internet. I kind of agree with her, and there's some reasons. She's big, she's politician type. What she says at some point is she describes how unexpectedly hard it was to do the kind of job I wanted to do, fill in the blank, and to be the kind of parent I wanted to be. And that's why you can't have it all. Notice the use of the word want. I would argue that you can't have it all if you have unrealistic expectations of what you can accomplish at work and at home, okay? That if you want to have the most publications and get the most recognition and get the biggest grant and be competitive and do all those things at home and be involved and be around, okay, for your kids, you can't. Something has to give. And so until we adjust our expectations of what we can accomplish, we're always going to feel like our performance at home and at work is substandard. And I've been there, and it's not a good feeling. So, you evolve out of that when you start to adjust your expectations. And I can't tell you that now. You have to discover it for yourself. I think you can't have it all, but you can have enough. You can have a rewarding career and a great family. And I guess my wisdom, for what it's worth, is don't compare yourself to others. It will kick you in the butt every time, okay? There's always someone at work who can work 24/7 and publish a lot, and maybe they do it effortlessly, or maybe they just don't have a life. And then meanwhile, I've got someone down the street who's a stay-at-home mom who's teaching her kids Chinese and has a clean house, oh my God, and and does craft projects and manages to get them to eat 7 organic fruits and vegetables every day. And I'm like, oh God, so. You can't compare yourself to other people. So have realistic expectations. Find a place or people that support you. And the question is it easier being at a teaching school? I know people really want the answer to that question, and I would say no. It doesn't matter where you are. It matters the culture of your department, your colleagues, the administrators, your direct supervisors. You have to find a place that you think will support you. And the second thing, and learn to say no and to prioritize. Okay? So this all begs the question of is there a perfect job? And I would say there isn't. The thing that will help it to be a good job is to find a job that allows you to do what you enjoy. You're going to work 60 hours a week in the beginning no matter what. It just depends whether you want to spend 60 hours a week writing grants or 60 hours a week grading papers and teaching. So you have to figure out what you're passionate about. So know your passion. Figure it out. My passion is teaching. Okay, I teach in the field. I have labs every single week. And when I say teaching, I mean teaching in the class, teaching in the lab, teaching in the field, teaching my research students. I do a lot of research, but it's with undergraduates. Totally different ballgame. Do I love it every day? No. But what I realized is it motivates me, it excites me, and it keeps me working hard. I realized that I'll stay up till 2 in the morning writing a lecture, or coming up with a really super awesome case study and a jigsaw for my students to do. I never stood up till 2 in the morning, I'm sorry, Dan, writing papers or writing grants. It didn't make me want to work quite that hard. And so that's when I realized I had made the right choice for me. So the differences between the programs, I'm going to hope to wrap up here at 4 o'clock exactly. The benefits of a smaller teaching school, you get to teach more. If you like teaching more, it's a benefit. I love teaching. Yes, the research expectations are much more modest. And what I mean by that is you don't have to write big grants to support your graduate students. In fact, they don't care if I write grants at all. If I can do my research cheap, cool, you know? And there isn't a lot of pressure to publish, okay? There's some expectation that you will, but at a very modest rate. You are, however, expected to do research all the time, and a lot of it with a lot of students, undergraduate students. And that presents its own set of challenges. It does potentially give you a little more flexibility in the summer with family. If you don't have graduate students, you can sort of arrange your time a little differently. The costs. You have to teach more. 12 credits I teach. Some schools are 15. That's 3 to 4 classes. There's tremendous expectation for departmental work, committee work, and university-wide committee work. The fact of the matter is, you know, in all honesty, you saw the data. I've made all my figures and graphs. I know my story. I'm set. All I have to do is write that paper. I won't look at it till next summer because I'm too busy, or Christmas, I guess, Christmas. So you don't have a lot of time. What that means is your productivity in terms of publication will diminish. If you can live with that, great. If you can't, then you're going to have a hard time. It means potentially you're going to have a harder time getting the grants or getting your collaborators at other institutions sort of throw you projects or co-author you or put you on grants, because ultimately they might not see you as someone who can turn things around really quickly, is what I've found. So ultimately you might be going, yeah, this is great, I don't want to go do the publish or perish thing, right? But I would warn you that most of you are you're coming out of an R1, you're coming out of a research culture. And even if you think that's what you want, you might find yourself sort of chomping at the bit that you don't really have the infrastructure, support, and money to do the kind of research that you've been trained to do. I'm just wanting you to recognize that there are trade-offs there, and you have to sort of think about what you're going to lose and what you're going to gain. So there are my research students from last summer. Out in the woods. If you want to apply for one of these jobs, here are my 4 tips. Get teaching experience. It'll help you get the job, and it'll help you survive your first few years when you have to do it all from scratch. It's a lot of work. Learn how to teach. I described what it means to learn how to teach. It's pretty rare. It helped me get the job as well. But I also, here's my number one thing. I think if you're at a teaching school, you're going to enjoy it. And you're going to love it for longer if you actually see your classroom as a place to experiment. If you do different things and try different things and it's fun and it's exciting. I think I'm having more fun than the people who walk in, turn on the PowerPoint, give a lecture, and walk out. That gets really boring after a while. So if you learn how to teach, you're going to be engaged and see it as a fulfilling career over time. And so find ways to do that. ESA meetings have tons of workshops. Take advantage of them. Maintain collaborations for your research sake with people at other schools. They're sources of cooperation, of ideas, of support, potentially of grants. Usually you're in a bubble. I'm the only forest ecologist. I can't have a conversation with anyone about what I do, but I could call Dan and talk to him about what I do. And ultimately, rethink your research. So this is my last point. Rethink your research a little bit. I think we walk out of a PhD being somewhat narrowly trained to ask specific questions in specific systems. And suddenly you're working with undergraduates who might have 2 hours a week or a couple weeks in the summer to do research, and they have no training and very little background. And so you might have to think of new study organisms and new places to go, and heck, hey, they don't even know how to drive yet, okay? Or they don't have cars, seriously. And you really have to rethink the kind of research you're doing. And you also have to take your big questions and be able to break them down into tiny little bite-sized pieces that they can manage. And then you can start to succeed. The reason I'm telling you that, whether or not you're going to do it, is it'll help you, but also if you are applying to one of these jobs, that is what your research statement should say. Don't tell me how awe-inspiring and sexy and earth-shattering your research is, because guess what? We don't care. What we care about is whether or not you can take that research that you've been doing at that big R1, which we are not, and you can make it understandable and feasible and manageable for our undergraduates, and you're going to do it with numbers. You know, list the projects you could do. Here are 6 projects I could do, here's the number of students I could do them in, and here's how long it would take me to do them. You'll get the job, or you'll get an interview. Okay, that's what schools, the #1 thing in undergraduate education is undergraduate research. The #1 high impact practice. This is what NSF cares about, this is what everyone cares about. You have to be able to do undergraduate research. It's the first thing we look at in our candidates, and we hired 2 last year. So, that's it. I hope the 2 minutes was not too terrible.
Questions for Suzie about anything?
About anything. And you please, you know, there's lunch, there's dinner, there's other opportunities. I also have lots of time during the day tomorrow, so maybe now that you realize I'm not super scary, if you want to talk to me about anything I talked about, I'm really happy. I'm here all day. So, other questions? Don't let Dan ask the first one, please. Yes?
I was wondering if you could give us some examples of the projects you've worked on with your undergrad.
Sure. They don't know what a model is. I'm not doing any modeling with them. So all of this work I've continued on my own, for example. I'm getting into old growth restoration. So I sort of found a project that works for me with the Nature Conservancy and the Bureau of Forestry. Immediately you have to find your contacts, you know, the agencies and people in the area that can get you going. And then what I'm doing is I am actually opting to not have them do a million different little projects, but in forest ecology, you have the benefit of being able to have 12 of them go out in the woods and measure a bunch of stuff for you. And that is experience, and that's valuable. And then what I'll do is I'll pull out little pieces of the data. So, hey, let's just look at the understory cover in light. It is not rocket science. It's not going to get published. But I'll give it to a sophomore or junior, and I'll say, hey, why don't you spend your fall analyzing this? And let's make a poster, and we'll present it at our little local regional conference. It's one step up from science fair projects in high school, and one step down from a very small master's sort of non-thesis type project. So that's one example. You know, I have a honors student who, because I'm doing this big project, I said, oh, hey, let's see not just what's coming up in our different restoration treatments, but what would come up. So we pulled out some soil, and she's doing a little germination study in the lab to see what sorts of tree species are coming up. Mixed success on that one. So, you know, I'm still trying to feed them into my research program and have them do things that ultimately will be helpful to me. Some faculty really create, you know, 12 different little projects, and they're going crazy, and none of it will ever lead to real research. But I want to keep being a scientist. Some people really go the other way when they get to these schools, and they just do science for the sake of doing it with students. And I still want to maintain my research. It's really important to me. So I'm trying to plug them in with smaller pieces, if that makes sense. And I could probably give you more specifics, but does that answer your question? You know, woods are great because you can just take them out and say, okay, measure everything, measure, mapping, and that takes a lot of time. Give them a compass and tell them to map everything. Yeah, so, Tom.
So your control group in one of your studies looked like you know, a mass of trees. Was it just 1 or 2 or 3 species, or because your models were 1 or 2 or 3 species? I was going to ask, what happens when you get to 10, 12 species and understory?
Yeah. That's a great question. You know, in Hawaii, we could work with multiple species. We were separating the 2 species from one another. In this system, I actually didn't need to do multiple species because it's almost all red pine. More than 95% red pine, scattering of, you know, an occasional beech or birch or something. So I pretty much could drop species out of the model. And we were thinning the understory. We actually, I mean, it's really much more complicated. Split plot design, half of it we left the understory, half of it we removed. And what we're seeing is a lot of preemption by the shrubs in the understory layer for resources. So the seedlings aren't growing where we didn't remove the understory. I'm working now with a 6 species system, and I'm really trying to separate all 6 species in those models. And I'm actually thinking in other ways about stuff like a diversity index. Can we model sort of the the diversity or maybe the evenness. You know, as the neighborhood evenness changes, are we changing competitive interactions? As the neighborhood diversity changes, can we model competitive interactions? Or grouping the species. In Brazil, we were looking at maybe separating, you know, shade intolerant from shade tolerant neighbors. So you can lump them and classify them if species don't show up. So I'm trying to help the Forest Service figure out why black cherry is declining. Because we love black cherry, it usually grows, and when we make a lot of money on it and it's not doing so well, it's getting outcompeted by maple and birch. So I'm trying to develop species-specific models to look at how a mix of species on the Allegheny are competing. Does that kind of answer your question? It is doable, it just gets more and more complicated. Yeah?
I'm coming back to your students. If you plan to stay where you are for a long time, one of the things that might be really cool is to have them help you develop long-term monitoring data.
Exactly.
And that helps not only you and the students get an idea of your longevity and change over time, but it would help the larger community.
Exactly. So one of them is in the state forest, where I really have established this huge treatment and fenced and unfenced and deer and no deer, and I'm getting them to do baseline established measurements, and I'm going to repeat. I've done them in 3 summers now. And the other is just there's woods on campus. You know, take advantage of. No one's ever studied them. Down behind the president's house, we went down there, and I said, okay, let's set up a bunch of permanent monitoring sites. No real clear objective or hypothesis yet, but we're measuring stuff. I should be careful. This is going onto the internet, isn't it? No, but the point is I think, you know, I can take my forest ecology class down there, and once a fall, we can collect a new set of measurements, and over time, I know how forests work. There is going to be usable data there, and I can start to have them see what's emerging. So absolutely, that's exactly how you can think about it with undergraduates. Yeah?
When you were evaluating your models, did you look for interactions of light with the different nutrient amounts?
No, because at that point I ran out of power with my data set. So, you know, you can only add so many parameters for a given sample size, and I was running into a problem there. So, for instance, I could have just added nutrients, right? Nutrients are affecting growth straight up. But I couldn't put that in and the interaction in. And ultimately, I don't really care if it tells me that they're growing a little more with more of the nutrient as a straight relationship. I cared more about getting at the interaction. So with that kind of modeling, you sort of have to choose which questions you want to ask. Does it mean I'm ultimately explaining everything about growth I could explain? No, but I'm actually just targeting specific hypotheses, which is sort of the approach with that. Does that answer your question? Yeah, I did try a bunch of other things that I'm not showing you, but yeah, those are the interesting ones. Yeah?
Kind of related about your relationship with control. I'm not sure if your control is the natural forest.
Yes.
It seems like even if you have experienced quite a lot of variability in natural forest, I would read it as like the light, the use efficiency of light is always optimal. So, you know, the plants don't spare any light, they use all of it, even when they are very patchy and diverse, whereas the manipulated patchness are a completely different shape from the natural use of light from the forest. So I'm wondering whether your reasoning really applies to manage environment, and you can inform the management and patchness and stuff, but doesn't apply too much to the natural variability in forests where no matter what, the light is very efficient.
Well, I would argue that it's not. I mean, I think I'm not totally sure I'm understanding your question, but
you have your thing, the blue spot,
it's flat.
All across.
Right. Once you get into a certain level of density, there's not a lot of variation in light, because at that point it's getting used by that full canopy and multiple layers there. This is also a 100-year-old forest that is actually somewhat homogeneous. It was a plantation originally. So I was surprised that there was as much variation as there was in a natural system where you have, you know, the heterogeneity we'd like to see and gaps and openings, and is light going to be used equally across a natural forest with natural variation? I don't think so. I mean, I would argue that it's not. So I think you could use this to look at different types of heterogeneity in natural systems. And I also think what's interesting is that you have to think about the type of forest you're in. And I couldn't really get into that, but you know, if you have a species which is got a really high leaf area and a really broad crown shape, right? It's going to use things more efficiently. It's going to shift that curve to the left. you're going to have a really steep drop off of resources because you're getting preemption and then a long plateau. If you have a forest type that has low leaf areas and sort of narrow crown architecture, suddenly, right, I mean, this is sort of what I was talking to you about with efficiency, you're shifting to the right because they're not really preempting those resources very, very quickly. So that was sort of a digression.